So thank you once again, and welcome session is done, and now we are moving to the keynotes. So the keynotes, the first session of keynotes is going to be about advancing data sciences and infrastructure, EOS perspective and innovation. And the first one, just in a minute, it's going to be about setting up an infrastructure landscape for human exposome research. And it is not going to be given by another person, that Professor Jana Klánová from the Research Institute Recetox at Masaryk University, the creator of IRIN infrastructure, and a person that once told me that it's lifelong creator of databases, you know. So I am really happy, and I'm looking forward for your presentation to show us how this is done within the exposome research. So dear professor, welcome her on the stage, please. Thank you. And the floor is yours.
Thank you for your nice introduction, and good morning, everyone. Happy to see so many people interested in data and EOS here. I just came home from Washington from the event called Exposome Moonshot Forum that was organized by the Johns Hopkins University at the Bloomberg Center with the hope of starting the global exposome project. This is just the flyer that was attracting the attention of the attendees for the workshop, saying that the huge human genome project produced a lot of data, but in reality did not fulfill the hopes, because it looks like only about 10% of the diseases can be fully explained by the genomic data, while we have all the other influences that were not considered. And if we really want to move forward with the precision medicine and public health in general, we need to set up something like a human exposome project. They brought together a very broad audience, representatives of the European Commission and the new leadership of NIH, many NIH institutes, European institutions, people from Japan, Asia, Africa, South America, to discuss. It was really a forum, so there was a lot of panels and discussions on how to move forward, what kind of working groups we need, how to set up the global coordination of such initiative, what should be the governments, what are the technologies. There were a whole lot of innovation companies and big vendors producing technologies that were also involved in the discussion. Of course, things like artificial intelligence and data were also discussed. Still, my feeling was that very few people understood the challenge that is behind it, because we see how we are fighting only the national situation, the European situation. And then you can imagine if we would want to try to bring together data globally. There was a lot of presentation on existing resources, like various human longitudinal cohorts across the world. There are hundreds of them. There is a lot of genomic data, exposure data, hazard data, but how you bring them together. So I should step back and say first, what does it mean, the word exposome? We celebrate 20th anniversary, actually, because Chris Wild, who is a former director of the European Cancer Research Institute, from his long-term observations 20 years ago already gave the presentation saying that we need something more than genome, that we also need to describe the non-genetic complement of the genome. And it took us 20 years to move where we are, and I don't think we are very far. At the beginning, there was a long discussion on the external and internal part of the exposome, because there are two ways how we can characterize what is happening to our body: by describing the external environment, so knowing something about the quality of water and food and air we are breathing, but then also describing the internal situation in the body, which is a measurement of the body fluids. We can measure various exposure agents, but also proteins or metabolites and so on. Later on, this definition was even broader, and this picture shows really the connection between the genome and exposome. Our body, which means phenome, is affected by the genes that we were born with, but also with a variety of factors that are together working on all the levels. So in reality, we should develop something like functional exposomics, because functional genomic describes how the genome is translated into transcriptome, proteome, metabolome, microbiome, and so on. And in reality, each of those levels is affected by the external environment. So the functional genome adds additional complexity. But then we also have to understand that it's a very dynamic part of the whole picture, because we are born with a genome that doesn't change very much. But then throughout our life course, the living environment changes a lot. Our psychological state, economic situation, environment, food. So it means ideally we need to bring the longitudinal element and search for the data that are longitudinal. This busy picture shows the long history of these 20 years of the exposomic research. It started very slowly, first 5 years, nothing was happening. Then both Europe and US starting opening the exposome programs, asking scientists what it is and how this can be implemented, which generated with some 12 European large framework programs addressing various parts of the exposome, many US programs. Until in 2017, we started thinking about the idea of building the underlying research infrastructure. Already in the S3 roadmap in 2018, there was a gap identified saying that we should really develop the infrastructure for the human exposome, because there was nothing that would bring together environmental and health part of the infrastructural domains. It was even worse because we see the chemical pollution as one of the important elements of the overall environmental crisis, but there was no infrastructure that would address it, neither environmental or health infrastructure. This is why we in 2020 submitted the application for the new European S3 infrastructure called EIRIN, which is the abbreviation for Environmental Impact Risk Assessment. And then in 2021, this research infrastructure was brought or prioritized to the European roadmap. These are the pictures taken from the European roadmap showing that EIRIN was pretty much translated into both domains, and it's very relevant for various subdomains because it brings together air quality, water quality, terrestrial infrastructures, but also health, clinical studies, biobanks, and so on, which of course doesn't make our life very easy. It's much easier to build a narrow infrastructure that is specialized on certain type of data than something that is really interconnected to many different domains. What does it mean to build this area of research and the research infrastructures? These are a couple of points that were identified in the large exposome conference that was already organized back in 2022, saying that we need to establish the standardized and robust measurement methods. And then everything else is pretty much about the data sharing, data repositories, harmonizing data, merging data, public health interventions, and so on. So very often I hear the question, so is the exposome science only the data science? Are you only talking about bringing all data from different domains together and analyze them jointly, preferably with AI? The answer is no, because it all starts with the quality of the data. So we really first have to think about the bigger framework and how those elements fit together, and then make sure that we at least start collecting the harmonized, standardized data. And then also, because the value of all data is very high, to also work on the harmonization of the existing data whenever possible. So if we go back to our picture of internal and external component of the exposome, really the challenge of building the exposomic infrastructure is to invest especially into high-resolution mass spectrometry for bringing together data on environmental chemicals, but also the biological perturbations, epigenomics, genomics, and all this information, and be able to wrap them up in the package that can be used in different population and clinical studies. So really developing something like the exposome piece that would be offered to the people who are running longitudinal cohorts of healthy people, but also the clinical studies. So this is how the whole concept of the IRIN research infrastructure was built. It is based on 6 different pillars. 3 of them are experimental, 3 of them are more data-driven. The first 3 are laboratory capacities for measurement of the exposures, so it means especially the exogenous chemicals. The second one is much more looking into endogenous chemicals. The third one is about toxicology. Because if we are assessing the exposures to chemicals, we also need to know something about the hazards that are connected in order to be able to assess both environmental and human health impact. And then 3 sources of the data. One of them is information about the human data, which is not only coming from the health programs, monitoring programs, clinical studies, but also from the biobanks and existing data repositories. Of course, the biggest challenge would be connection with the health data. The second pillar is more on the environmental data coming from the environmental monitoring of water, air, food, feed, and such. And then the last one is about bringing data together and building all types of models, prediction models, spatial models, the pharmacokinetic models predicting the behavior of the chemicals in the body and such. I should say why we at Masaryk University decided to build such research infrastructure, because we already had a long history to build on. Because the Czech component of IRIN was in fact prioritized in the Czech national roadmap already back in 2011. We started building various layers of this infrastructure gradually. So we started with the laboratories that we currently have a large capacity of accredited trace laboratories, but also laboratories for the high resolution non-target assessment of both environmental and biological samples. Then later on, we added the monitoring studies. We are running the big monitoring networks globally. We, 10 years ago, continued with building the population studies. We have 3 generations of the young birth cohort, grown-up birth cohort, and aging cohort, and also some national seroprevalence studies. And then, of course, underlying data structure, and not only repositories, but the systems that would allow you to analyze, interpret, and visualize the data. And this is what we were building on. Another advantage that we have is that we have long-term collaboration with the policymakers. And as you know, the research infrastructures, very often in the last years, we are discussing about their social impact and whether they are also delivering something to the society on the top of the advantages they are providing to the scientists. Since 2008, we serve as a collaborating center of United Nations Environment Program, and we are giving a support to the international conventions protecting human health from the environmental hazards, like Stockholm Convention on POPs or the Minamata Convention on Mercury. And for them, we are developing the data systems. So these are the pictures taken from our Genesis and GMP visualization tools. The GMP is really the tool that UNEP is using for collecting all data about the human exposures and environmental exposures globally, and we are responsible for their visualization and update. And this is still simple because here we are talking about the targeted data, but I said that we are now interested not only in the targeted data on exposures to the specific number of exogenous chemicals, but we are interested also more in biochemical responses. So measuring the metabolomic, proteomic, targeted and non-targeted data, linking it to the biochemical measurements. And as soon as we come to high resolution profiling, it's the non-target work that is generating the massive volumes of the data comparable with the genomic data. And then it means that we require much bigger storage capacities, but also supporting capacities and data processing pipelines that could make sense of the data. Because we need to understand that genomic project in this sense was much easier. You have 4 building blocks, you read it once, you have the human sequence. Part of the high resolution mass spec data means we are working with thousands and hundreds of thousands of unknowns that can be explored later. So we really have to make sure that we are collecting data in the proper way and store them so they are really not only findable, but reusable for the future exploration once we know more about the chemicals we are looking for. So the IRIN concept of the exposomic means that we have to start from the beginning of the chain. So we are investing a lot into developing and sharing the standard operational procedures that are all publicly available, that all our spectral libraries are being shared. And I think we are still the only ones on the global market that are doing that. We are investing in close collaboration with the Elixir infrastructure and Galaxy community into building public data processing pipelines in the Galaxy environment that, again, would make sure that data are treated in the standardized way. And of course, this also means linking available data that are currently in databases that are anything but fair. This means that we need to think about how we will connect the whole this domain to other existing domains. So it means we need to collaborate not only among the environmental institutes and infrastructures, but for us, it is very important to have a collaboration with EMBL, EBI, for example, and other European structures. I already mentioned the Elixir infrastructure, EGI and EGA. This is something that we are currently exploring, trying to find out how much of these capacities we can actually use and how much needs to be developed specifically for this purpose. And this is also why we were quite active in the development of the national EOSC project, especially now building the OS2 project, where we took responsibility for the development of repositories for data on chemical exposures and their effects. And this is just the very brief structure showing in which repositories we want to invest. I already spoke in the first pillar that we currently have the family of the Genesis databases for the targeted data. This is something we, of course, continue developing, but we really want to turn it into the fully accessible and fair database that would serve also to other projects, not to UNEP and WHO only. But then the second repository is the repository for the metabolomic data. And this is the big space that we really need, but it is especially important to develop correctly the metadata structure because none of existing metabolomic databases is really reusable. This is something that we are trying to do with EMBL EBI to make sure that what we develop can be adopted. And it's a question of this for discussion whether we should develop it here, whether we should collaborate with EBI and let them develop it centrally, or whether we should set up much closer collaboration with existing human genome projects that are working on developing the federated European structure for the genomic data and encourage our local partners to team up with the local people responsible for human genome projects. I already spoke about the toxicology. This is completely undeveloped at the European and I'm afraid global level, really centralized databases for the genomic data. And the last part are more environmental data because, you know, to describe your living environment, you also need a lot of data from satellites, from remote sensing, you know, about the physical environment and such. So we shouldn't look only at the human body, but also at the data existing outside. And as I said, this brings together the data that are currently stored in different places and supported by many existing European infrastructures. So just to give you an idea, we are talking about the data about air pollution and noise and light pollution and pollen and all kinds of the traffic in the city. It, at the same time, brings a lot of opportunities to also work at a local level. And this is actually the fourth repository that we are building. I even hesitate to call it a repository. It's really bringing together different GIS letters so you can pinpoint the working place or living place of the individual and describe the cumulative risk that is based on the external environment. And this is something what we play with when we are talking about Brno Living Lab, just taking advantage of our pretty much 40 years of the activity in this area, being able to collect a lot of population data, a lot of environmental data, close collaboration with the city and the region, building the kind of digital twin Brno Living Lab that would allow the citizens of the city to benefit from the data that were already collected in the area. Of course, we are building on important pillars. There is an existing international human exposome project currently supported by the European Commission that is only CSA project, bringing us together with all relevant partners to discuss how to move next. I already said that similar project has been recently granted to Columbia University in the US to coordinate the whole structure in the United States. And then we are also supported by the European Partnership Park, which is a 40 million euro investment into extracting the data that can be used for the European chemical policies. So we are making sure that everything we are doing can also be translated into the policy making and used by the regional authorities, governments, and common citizens. So this is the quick overview of where the exposomic science is today. Thank you very much for your attention and happy to answer any questions.
So once again, thank you, dear professor, for the presentation. And we have time for two questions. And as I promised, one from the audience and one from the Slido. So who has the question from the audience? And I can see over there, Jiří Vondrášek. So please, Domcha, if you can. No, please wait for the online, the online participants can hear it. Thank you.
So the interoperability is something which you, I think, solve on the everyday basis. So you can just answer. I don't know.
You have to start somewhere always. And I'm a lab person, so I'm always trying to start with the high quality of the data. And this is really hard of Irene, to start from building the standardized capacities. And even that is a challenge because people in general are not willing to share, and we all know that half of the data being published is really a crap, and it's not well documented, and it's not reproducible, and you can't do anything with it. Regarding the connections, you have to start with the significant cohorts. So the plan is to really start with a very few large cohorts that already have much of this data available and connected, and start adding layers. Actually, when I was sitting and listening to the moonshot, it is growing way too big, and it's becoming the science of everything. And I think this is the biggest danger, because we really need to be careful to make sure that we are taking one step after another, building something solid. So not to get lost in the huge ideas about the moonshot. So our approach really is starting with the laboratories, working on the catalog of the cohorts, selecting few representative cohorts, building the pilot cases on, bringing them together, And many of these initiatives already happened during the previous European or US project. So it's just important to give it some sustainability, which is the major, that European projects, you would bring together consortium, work together for a couple of years. Once the funding is gone, everything is gone, and you start all over. So this is really about providing a longitudinal platform.
Okay, thank you very much for the question. And are there any questions via Slido? There are not. So we can ask another question in the audience here in the room. So what about any other question? We have time. Okay, so I have a question. What's the biggest barrier? Quality people, finance, or infrastructure? What's the biggest challenge?
I always think that the biggest challenge are people, because you really, and we just discussed it before the beginning with Honza, the people who have both the scientific understanding and still managerial skills and the will to do something like that, because many people just work in the silo, they are hunting their excellent publications, they wouldn't be willing to step outside their comfort zone.
Okay, thank you very much. So another question from the audience. This is really the large-scale use of interoperability in life, so use the opportunity. Anything on Slido? Domcha? No? Anyone? Okay, so with that, thank you once again, and enjoy the rest of the conference.
Thank you very much.